Okay, perfect. Let's do that. Let's continue. All right. So we have a complexity of the cells of the immune system. And there is a common myeloid progenitor and then common lymphoid progenitor. So if you can think of this, how the stem cell, hematopoietic stem cell, right? So the dedicated immune, so to speak, and blood stem cell develops, it has two major arms. That's what people know for sure now. There is a common myeloid progenitor, common lymphoid progenitor too. And it is interesting to see how this, in some sense, mimics observations made by Ilya Medzhitov and Paul Ehrlich, right? Because the plasma cells producing the antibodies are really part of this common lymphoid progenitor, lymphoid cells, while the macrophages and the cellular immunity concept by Ilya Medzhitov was really originating by studying the cells of the common myeloid progenitors of the myeloid origins. So oftentimes in immunology, these cells, T cells and B cells, are referred to as lymphoid cells, and the other ones are referred to as myeloid cells, macrophages and dendritic cells, for example. And then the split here is actually that there are two immune systems, two components of the immune system, the adaptive immune system and the innate immune system. The way to distinguish it is actually the easiest and the most well-defined way is by looking at the evolutionary perspective. And then the evolutionary perspective is that innate immune cells, such as macrophages and so on, has been present in great many organisms for a very long time. However, the adaptive immune, such as T-cell receptor and B-cell receptor carrying cells, they only appeared from what's referred to as jawed fish stage and later on. So these cells, T cells and B cell mediated immunity, is only later development in the immune response against pathogen. The other aspect of the innate and adaptive immunity is the split in the time of the response, right? And so the first responders are the innate cells. So the macrophages, dendritic cells, neutrophils, they are the ones mounting immediate, within days or even hours, response to the pathogens, producing cytokines and so on. And then at about day 7, the adaptive, the T cell, B cell mediated, particularly T cell mediated immunity kicks in. You might know the expression that when you get a cold, if you take a medicine, you'll be sick for 7 days, but if you don't take, you'll be sick for a week. And the expression comes, the timeline comes from the fact that it is about on day 7 when you have this major T cell immunity kicking in, which is very specific and very strong. Now, the macrophages themselves, they are actually present at almost all of the organisms of the body, and then they survey the environment at all times, even without the any specific immune signals. And for example, some of you might have tattoos. So the reason why tattoos are staying stable, tattoo ink is staying stable under the skin, is because it is phagocytosed by the local tissue resident macrophage. So those are your tattoo cells, and you can color them like this, and you'll see it's going to be. It's not just an urban legend. There is actually legitimate scientific publication discussing the tattoo persistence and the role of the macrophages in these cells. And but most importantly, when you cut the skin, for example, and some of the microbes get under your skin, then the resident macrophages will sense that there is a microbial surveillance, and so they will sense the danger and will start activating. So this is one of the most important parts of the macrophage response, is the activation, and inflammation is actually triggered. So basically, that is triggered by bacteria, is key for sensing the cells in the tissue macrophages. So basically, the macrophages engulf, phagocytose the microbes, and then they have various mechanisms inside the cells, such as the reactive oxygen species or very strong lytic enzymes. These mechanisms will destroy the microbes typically, and then go as much as actually even release the destroyed particles into the space. Now, what is the question here? What you might have picked up on here is that macrophages should be able to somehow recognize the fact that there are microbes around, right? And furthermore, this is also suggestive because evolutionary T cells and B cells were not a part of the immune system for a long time. So which means that, again, innate immune system, macrophages, should be able to have some basic recognition mechanisms, evolutionary conserved recognition mechanisms that would be showing the response to the indicating the response to the presence of the pathogens, showing the fact that some foreign microbial patterns are present. And so the person who put this hypothesis forward in this kind of concise manner was very famous immunologist, Charles Janeway. He worked at Yale at that time in the early 90s, well, throughout 90s, in fact. And so he has put forward the hypothesis that there is a receptor which can recognize bacterial and more generally microbial patterns in a general way, and then trigger the activation pattern of the macrophages. Now, this receptor is evolutionarily conserved, and so people were looking at this not only in macrophages but also in response to microbes from Drosophila flies and so on. And so one of the major components, typical molecule associated with the bacterial walls, is something called lipopolysaccharide. So this is structure that is present in almost all gram-negative bacteria. And so the macrophages are notoriously well in recognizing the presence of LPS. And they do it through this toll-like receptor 4 agonist. So TLR4 agonist, which is the TLR4 is a receptor, toll-like receptor, which was first found in the fruit fly, in Drosophila. So Janeway was also the first person, well, he's not the first person to write the book, but he's written this very nice conceptual textbook, which was first edition in 1994, Janeway's Immunobiology textbook, which has since become a Bible and a classical textbook for anybody studying immunobiology. And it has survived 10 or 11 now edition. This is from, I think, 10th edition is current one, and so this is from last year picture. This is a textbook of immunobiology that is present, whose author now, because Janeway passed away, the author is Ken Murphy, one of the leading encyclopedic knowledge scientists in immunology, and he also happens to be a colleague of ours, of mine in the Washington University Immunology department. And here is the paper from Janeway's lab that actually highlights the reports the discovery of the toll-like TLR4 signaling receptor that recognizes the LPS. And you can see that what they discover a homologue of the particular protein from Drosophila, which is able to activate the immune response, and particularly by antigen-presenting cells, by the macrophages. You might recognize the name. You can see that the first author is actually Ruslan Medzhitov, one of the, I think, hold on, there is a question there. Is there a difference in time of antibody production between bacteria and viruses? Yes, there is, but I would not go even as far as to say difference between bacteria and viruses, but there is a major difference in major variance in various bacteria produce antibodies in different times, and various viruses produce antibodies in different times. So this is a very delicate play in the specific timings of antibody production. And of course, every pathogen is trying to delay to make the antibody production as late as possible. So I wouldn't kind of just put them into the buckets of the bacteria versus viruses there. Okay. All right. So then this is the paper by Ruslan Medzhitov. There is a beautiful, I think 2 or 3 YouTube talks. They are not very long, they're just 20-21 minutes, where he describes a human homolog discovery and his path. He studied at the Moscow State. He was born, I think, in Tashkent. Was studied in Moscow State University, and then he wrote mail, I think, at that point to Charlie Janeway, and Charles Janeway has invited him to be a postdoc at Yale, and so he worked in his group. However, you know, unfortunately, Charlie Janeway would be a very worthy recipient of the Nobel Prize devoted to this topic. However, he passed away in 2003. But there are 3 worthy recipients of Bruce Beutler, Jules Hoffman, and Ralph Steinman, who are recipients of the prize in 2011 for the activation of the innate immunity, and particularly for discovery of dendritic cells and role in adaptive immunity. Sadly, Charlie Janeway passed the age younger than typically scientists do. So Sasha, he had, in addition to Ruslan Medzhitov, he had Sasha Rudensky and Sasha Chervonsky, 2 other Russian origin postdocs in his lab, who both went on to become a very famous immunologist. And Alexander Rudensky has been one of the discoverers of FOXP3 and regulatory T-cells. And the other interesting aspect of this prize is that it is one of the only perhaps prize. As you know, the Nobel Prize is never given posthumously, meaning that the person, it only is given to the live people. And it turns out that even though the Ralph Steinman passed away on September 30, just before the prize technically itself was announced by October 3. On October 3. So, you know, it's technically one of the few prizes that has been given if not the only one, after the person was known to be dead. So that's a bit of the story of the TLR receptor that culminated in the corresponding Nobel Prize discovery. And so now we can think of kind of a little bit, go over the key role of the macrophages. And one of the immediate roles is the destruction of the pathogen, right? And so the way how it initiates by the macrophage recognizing the microbial pattern, right? So when macrophage recognizes that there is a true receptor, TLR4, it binds strongly to the lipopolysaccharide, LPS, on the surface of bacteria. This is when the activation of the macrophage starts, meaning that the signaling cascade propagates, the major transcriptional program, which you will see on Wednesday and Friday, goes on, and with the various aspects of this program. So some of the aspects regulate the way how the microbes are being engulfed and how they are being destroyed. One of the mechanisms is, for example, macrophages has these granules with lysosome, with very strong enzymes, which then merge with the vacuoles that contain the microbes, and then This digests, literally digests the microbial patterns, microbial proteins, etc. And then all you have done is microbial debris, which are then can be released by exocytosis. Now, what is happening, not only this destruction process happening, but there is a number of the cytokines that are being produced, and which we'll get to. And so some of these cytokines, they have to be upregulated first on a transcriptional level. And so this is the list of the various genes that are being upregulated upon the enzyme, upon the macrophage activation. And so you'll get to see, again, in the bulk RNA sequencing, bulk microarray data, you'll get to see how these genes are going up quite literally in your hands, computationally at least. And what is these genes stand for? IL-6, IL-12, TNF, IL-1 beta. These are the proteins that will be released from the macrophages, and they will inform cells around macrophages that immune response should start. So not only immediate destruction of the pathogens is important, but also the other component that is very important, which is the activation and activation of the subsequent immune responses, and informing changing the local environment to the pro-inflammatory state. And so one of these aspects is the priming of the adaptive immune system here. So the way what macrophages and other antigen-presenting cells do, not only they try to destroy the pathogen, but they also start the adaptive immune system. And then this is where the process becomes very critical. So what is very interesting observation is that the T cells can recognize pathogens not in a random way, but only from the macrophages of the same genetic background. And we'll come back to that in a moment. But for now, what is critical for our understanding is that not in this phase here, macrophages are activating, trying to kill the virus or the bacteria, and then start producing cytokines. So the molecules such as IL-6, TNF, tumor necrosis factor, IL-6, and IL-12, and so on. The molecules that will create pro-inflammatory environment, and that they will inform other immune cells, such as T cells, to start activating and coming to the site of the inflammation. Now, this is a very important part, that not only evolutionary, there are two parts of this process that are happening, but also you have the time-wise two phases, one associated with the innate immunity, the other one associated with the adaptive immunity. And why are these two phases, and why are these two phases really critically important? Because, again, coming back to what we said initially, immune system is one, if not the only system, which can destroy, not only destroy pathogen, but it has a potential to destroy your own organism. Right? So it is a very, very strong and very powerful weapon, and it is very important to not to make sure that this weapon is not used to shoot yourself. And so the two phases here, they represent the balance between the strength of the response and specificity. So the innate immune response, because the TLR receptor, toll-like receptor, is very specific for the very general patterns of the bacterial wall component. Some of the cells, sometimes the activation can go aberrantly, can go wrong. And so the macrophages do not have a very strong ability. They have some, but not a very strong ability to clear the pathogen or destroy the cells of the organism. However, the T cells, they actually have a very powerful T cells and antibodies. They have a very powerful abilities to destroy the cells associated with the antigens. And for example, the reason we know it is, of course, that majority of the autoimmune diseases that we know are associated either with some aberrant antibody productions, such as some lupus diseases, etc., or with the appearance of the wrong T-cells that are start targeting your own self component. Some of the most known ones are things like celiac disease, which would start targeting particular peptide presented on your cells or other components of it. And so the way how this specificity is achieved is through the T and B cell antigen receptors. So the way how they work is a very unique manner, which again, we're not going to go through into details, but the T cells and B cells are the only cell types that can have DNA rearrangements after the birth of the organism, planned one. Of course, the tumor cells have plenty of the DNA rearrangements, but those are not planned. And the way how the antigen receptor in T cells is working is that in the B cells, it adjusts and changes itself to achieve the maximum compatibility or maximum recognition of the particular antigen. And once it's done, then it starts a very, very strong immune reaction that is very, very specific to a particular cell type or a particular antigen. And for our purposes, for the purposes of the next 3 weeks, we'll be focusing less on B cells and slightly more on the T cell mediated immunity. And as you can imagine, some of the things are actually can be very challenging, because when your receptor is mutating so quickly or is able to be so diverse, to change so quickly, then you should have a very particular system that interacts with this receptor. And so in people and in mice and in mammalian organisms, this system is known as TCR-pepMHC interaction. So on the T cell side, you have this T cell receptor with the parts of it that are very variable, while on the other sides, on macrophages, but also on other cells, you have the system which is called MHC, major histocompatibility molecule complex, which has not only the surface molecule itself, but also particular peptide in it. I think you might be a bit confused here or lost, because it is a very complex structure to arrange and rearrange. So for now, think of this as there is a T cell receptor, and then there is a very strange type of a receptor that interacts with the T cell receptor from the side of the, for example, macrophage or any other cell. It turns out, to add even more level of complexity, there is one major type of MHC presentation, which is called MHC class 1 presentation. But the reason why this complexity exists is because the many pathogens are intracellular. Right? When we discussed antibody floats in your blood, for example, can enter some different tissues and so on. But so antibodies against the viruses, what they cannot do, they cannot get inside the cell, right? And so if the pathogen, bacteria or the virus, are spending a lot of time outside the cell, then antibody can be very, very helpful, because it will opsonize the virus or the bacteria, and then macrophages will eat up the opsonized bacteria very quickly. Now, many times in fact, especially with the viruses, what the viruses do, they like to get inside the cells as quick as possible and spend as much as possible inside the cells. And the reason for this, of course, because viruses lack any proliferation machinery. Virus by itself cannot make more viruses. In order to make more viruses has to get inside the cells and use the machinery that exists in a host cell to make more viruses. So this is the very common typical scenario, and this is the reason why the T-cell mediated immunity exists. Because in a way, you can clear out the bacteria or viruses that are floating in the extracellular place, but you cannot clear out the viruses from inside the cells. And the approach that immune system chooses is that I'm going to kill the, if I see the infected cell, I'm going to kill the infected cell completely. So not just kill the virus, but kill the virus and the host cell. So this is the way, modus operandi. This is the way how the immune system works, and this is the reason why CD8 T-cell mediated immunity is critical. It helps to fight against various intracellular antigens. Okay, so when you have the intracellular pathogen, what is important is that you have somehow the idea of what is happening inside the cell without killing the cell, right? So you have to know whether the cell is infected or not before you decide to kill the cell or not. And this very specific system of TCR-pepMHC interaction, it provides exactly the mechanism. This complexity and the challenge of this situation is specifically due to this requirement that the cell should be able to somehow indicate to the T cell, any cell of the body, indicate to the T cells whether it is infected or not. And in fact, MHC class 1 molecules are expressed by all of the cells in the body. And yeah, so the basic mechanism is that the killer T cell with the antigen, with a specific antigen receptor. We haven't talked yet how this specificity is achieved, but the killer cell with a specific antigen receptor can, when it recognizes the presence of the pathogenic component in the cell, then what it does is just immediately kills the infected cell. The way it is achieved by, so what is happening is that once immune system T cell receives this signal that there is a pathogen, the very specific T cell receptor starts, the T cell bearing that T cell receptor starts proliferating very heavily, producing what is known as a T cell clone. So this is the T cell with all of the identical T cell receptors, which then carries out the killing. And once the virus is cleared from the system, when there is no more cells that indicate that they have a virus, then starts the contraction phase of the CD8 T cells, when they basically go into commit some kind of suicide, apoptosis of the cells. All right, so let's try to recap. I think what we'll do, we'll try to go recap, and then we'll take 5-minute break to kind of get, I think there is a question there. Yeah. Oksana?
I just wanted to ask, so do I understand it right that the only way to fight the intracellular antigen is to kill the whole cell?
Yeah, pretty much. Yeah. So it's when the virus takes a hostage of the of your host cell, then immune cells, they kill both the criminal and the hostage. They basically decide. Well, there is technically, I guess, one more way can be to try to encapsulate, to try to kind of isolate the cells, but that usually is not very productive. Some of these things can be, you know, like formation of granulomas. That's when the immune system cannot clear. Then it says, okay, somehow I'll try to isolate it and fix it. All right. Any other questions? We are getting into the kind of challenging, very non-linear aspects of the immunology here already, right? Okay, so let's try to recap just a little bit before we go into the 5-minute break, short one, that how things are working. Now, you have innate immune system and adaptive immune system, right? When you cut your finger, for example, the bacteria gets under the skin, and everywhere in your body, there are resident macrophages. Even in the brain, they are called microglia. There are everywhere present resident macrophages, which have the particular receptors called TLR receptors. TLR4 is one of the most famous ones. There are other, TLR3, 2, etc., that recognize some very stable bacterial or viral pathogenic patterns, something that is really generally shared between the most of the pathogens. So once the pathogen gets inside the inside your body, under the skin, local resident macrophages immediately feel it with the TLR4 receptor. They activate and they start doing two things. One, they start being very actively phagocytic, and two, they start which they phagocytose. They activate the machinery that helps killing the bacteria inside the macrophage. And then the second aspect is they start producing various cytokines, IL-6, TNF, IL-12, and inducing other molecules that are critical for the immune responses. Now, the activation of macrophages is immediate part, but then the other part is macrophages have a very limited ability to kill the cells, right? So they can clean up some cells. Usually when the cells are already dying, that's when macrophages clean them up. But they don't usually go and actively kill the cells of your own organism. But what the activated macrophages do, or myeloid cells do, they activate the signaling cascade that leads to the activation of the adaptive immune cells, particularly T cells. And T cells have this unique ability to recognize the antigens in a very specific manner. The reason is because the antigen receptor in T cells is very variable. In your body, there is many T cells, and each cell carries separate antigen receptor. So you have about 10 to the 11th T cells, and each one of them has individual T cell receptor. And only one or two of them will be specific to your bacteria that got in. It will recognize the peptide coming from bacteria presented on the surface of the MHC of the macrophage. Or if the virus infected the cell next to the macrophage, then macrophage itself will not be killing that cell, but the T cell will come in and will have to actually kill the cells that are infected in the site of the infection. So the T cells, they don't clear the bacteria that is floating around in the extracellular milieu. That is done by macrophages, that is done by the opsonization with antibodies and so on. But for the T cells, it really is focusing on finding the bad cells, the cells that are infected, and cleaning them up, because the infected cells is the major reservoir for the virus to proliferate. All right, and once this is achieved, then the T cell that has expanded, once it killed all of the cells that contain the virus, then it starts contracting, and basically most of these cells, they die off, and only few cells remain, which are the memory cells, which will be very useful cells for the next time you'll be infected with any similar type of the virus, because these cells will be able to respond to this situation. Okay, questions here? No questions so far? All right. Okay, so let's take a 5-minute break now here.